Okay, folks, so this is the last exercise we're doing today. Oh, oh, you need a plate. Okay, sorry. Let me get you a plate. Oh, that's too wet. That one's better. There you go. Okay. All right, folks, so we're working off of our handout, and now we are on page 5, folks. So again, I'm going to walk you through how to streak a plate to get isolated colonies, but the step-by-steps are on your handout here. Okay, so you always have that as a reference. Okay, folks, so let me see if I can do this methodically. So folks, what we first want to do, of course we need to label our fresh TSA plate, right? So I'm going to show you how I was taught to streak a plate, folks, and I still use it because it always works for me. If you've taken micro before, if you worked in a lab, and you have a different way to streak a plate to get isolated colonies, go for it, right? But I'll show you the way that always works for me. So the first thing, you guys, on the bottom of my plate, I want to divide it into quadrants, like it's a big pie. I'm going to divide it into 4 quarters or quadrants, okay? And you can use a wax pencil, folks, or a permanent marker, right? And then, folks, the technique I'm going to show you, we're going to streak quadrants in order, 1, 2, 3, and 4. But because I can't do things in mirror image, you're going to laugh at me. I have to turn my plate over as if I'm going to be looking at it while I'm streaking. And now, you guys, I'm going to number the quadrants going clockwise. So 1, 2, 3, and 4. And you guys, if you misnumber them, don't worry. This is just to help you try to keep track of which quadrant you've streaked already, which quadrant you're going to streak next. So, and you guys, again, for the movie, we'll try to do it up here on the board. So you guys, this would be my quadrants numbered as I'm looking down on it. Okay, so quadrant 1, 2, 3, and 4. Okay. So, of course, you guys, we need our clinical sample. So remember, we're using a pretend urine sample from a patient we think might be suffering with bacterial cystitis, a bacteria bladder infection. This is a midstream catch, right? So we know we're going to be harvesting part of the normal microbiome, you guys, that live on the mucosa. But what we're looking for specifically is a possible pathogen in the urine itself. So we're going to take the urine sample and spread it out over our 4 quadrants so we can get isolated colonies. So the different bacteria will form different colonies. We want to remember, you guys, that an isolated colony, our hypothesis is that all the members, all the cells in that colony, are descendants of one cell that landed on the surface of the plate. So consequently, they should be genetic clones. And thus, an isolated colony, a colony not touching another colony, should be a pure culture. And we're going to see this is going to be really powerful for us. Okay. So I need my urine sample, folks. Here's my urine sample from my patient, either patient pink or patient blue. I'm going to need a loop this time, you guys. I really would recommend you use a loop because we want the loop to glide over the surface of the auger. I think of it almost like ice skating, right? And for me, if I use a needle, it gouges the auger. Okay, if you gouge the auger, it's not a big deal, you guys, but it's just nice when the loop glides over the surface. And of course, folks, we're going to do aseptic technique. So what else do we need? Our Bacti incinerator, right? Because we're going to do an aseptic transfer. We don't want to contaminate with unwanted environmental microbes. Okay, and again, you guys, I'm so bad. I should be doing this in my full PPE, right? Okay, so I've got my plate labeled. In addition, you guys, of course, you want a good label with the patient's name, urine, your name, date, lab section. So we've got all that going. And then, folks, whenever you're going to streak a plate, if you have your microbes are in a liquid, it's always really wise to do what before you take your sample? Just gently mix it, right? Because a lot of times the microbes will settle to the bottom. So I would close the cap tightly, you guys, hold on to it, and just gently flick the bottom, right? And you'll probably see, oh my gosh, there's clumps of bacteria in the bottom there, right? Isn't that, ooh, these poor women. Okay, so I want to mix our urine really well. Good job, you guys. All right. And then, folks, I'm going to take my loop, and what do I need to do first, you guys, with my loop? Sterilize it, right? So remember, you guys, always by the end of the handle, because you'll burn yourself if you handle it up there. And we're only going to put half the wire inside the barrel. Count to 5. Everything should be incinerated, right? And now, folks, let's see here. I think I'd actually recommend, if you can, try to hold, I know it's like you need a prehensile tail here. Maybe try to hold your sterile loop in one hand, grab your urine culture in the other, and we're going to do the same thing, you guys, as we did with aseptic transfer into the big butts. Just going to hold the cap to the barrel 1 second. Grab, you really need a prehensile tail. Grab the cap in your pinky, remove the cap, try not to place the cap down, you'll contaminate it, and then just briefly warm the lips again, trying to get those warm air currents, pushing the microbes out. You guys, with liquids, the good thing is even if your loop is hot, if you insert it into the broth and it hisses and spits, there's still so many microbes in there. Just leave it there until it stops hissing and spitting, and there will still be enough microbes present. You won't kill them all, right, in a large volume of liquid. So you're going to insert your loop, right, all the way down, and then withdraw it. And again, you guys, we just warm the lips, put the cap on, barely loose, right, to vent. We're going to sit this back in our test tube rack, our urine. And you guys, the key is don't go back and get more urine. Our instinct is, that's not much urine. I'm going to go get several more loops. The key is only one loop full of urine, okay? I'll show you guys on the plate, and then we'll cartoon it on the board. So what I want to do is I want to spread that one loop full of urine over the surface of quadrant 1. And I want to use as much of the surface area as possible. So you guys, I'm going to start with my loop really close to the wall of the plate, and I'm going to spread or streak the bacteria over the surface of the plate and really tight, narrow little streak lines. And I'll show on the board what I mean. It's important, you guys, you don't go over the boundaries of your quadrant. You want to stay within your quadrant. Okay, so I've just streaked quadrant 1. I'm going to close this. And now, you guys, I want to get rid of all the millions of bacteria on my loop. So what am I going to do? Yeah. So you guys, between each quadrant, you're going to sterilize your loop. And this is the other thing folks forget to do. Okay, because we're trying to get the numbers of bacteria really low. And the only way we're going to do that is if we sterilize our loop between quadrants. Okay? Yeah, guys, let me just show you on the board. So you guys, I just went in, I got one loop full of urine, right? And now I'm going to streak it out using as much of the surface area of quadrant 1 as I can. And again, I want to be careful that I don't go over the boundary lines. I don't want to get into the other quadrants. Okay, and remember, you guys, one loop full urine only. Sometimes we make jokes and we say, have your partner hide your urine sample, right? So sometimes I just got to get one more loop full, just one loop full, okay? All right, folks, and then what we're going to do, we're going to sterilize our loop, and now we're going to drag just some of the bacteria from quadrant 1 to where? Quadrant 2. And you guys, this is just how I do it. Everybody kind of develops their own technique. I usually do like what I call 2 pulls, 2 streaks from quadrant 2 to quadrant 1, so 1, 2. And then, folks, I want to streak those bacteria out over the surface of quadrant 2 in just the same way. We're trying to make really tight streak lines, trying really hard not to go over the borders. Okay. So then I close my plate, and what should I do with my loop? Sterilize, right? So if in doubt, so we're going to sterilize. If in doubt, wash your hands and sterilize, right? And we should be careful, you guys, that when we're sterilizing, that we touch our loop to the uninoculated agar to make sure it's not too hot, because if we touch the bacteria on the surface of the agar with a hot loop, it will kill them. Okay, let me get another color for you guys. And then I'm just going to repeat this over and over again. So I'm going to pull some of the bacteria from quadrant 2 to quadrant 3, right? And I'm going to spread them out. Okay, close the plate. What do I do? Sterilize, right? Good. Cool, right? Cool. And then, you guys, historically, quadrant 4 used like great big 4 lightning bolts, right? So I'm going to pull 1, 2, and then I'm going to use great big like lightning bolts in quadrant 4. Close the plate. Really important, you guys, don't forget, sterilize right at the very end. Good? Okay. And then, folks, what we're going to do is we're going to tape the lids to the bottom, invert the plates, and put them at 30. And you guys, something that your colleagues came up with, which I thought was really smart, when we start doing metabolic tests, or you start doing the unknowns, there's going to be zillions of cans and plates in there, and it makes you crazy having to spend time trying to find your plates and cans. So what your colleagues, I thought this was really smart, what your colleagues developed was, if you have your well-marked can, where's a good place to put your plates? Underneath it, right? And that way you can spot your can and you'll know, boom, I know my plates are right there. Okay, and if they're taped together, lids to the bottom, it should be fine. Does this make sense? Okay, awesome, you guys. So remember, we're practicing. Don't stress out if you think you made a mistake. That's why we're here. Just so you know, you guys, on Thursday, we'll be looking at our TSA big butt slants. We'll be looking at our plates, and we'll also, you guys, do the follow-up on the airborne microbes. We're going to count total colonies, different varieties of colonies, and you'll practice describing 2 specific colonies. And if you guys, before you leave, just take a look at your plates and just, you know, kind of give thanks to your immune system, because when you see what we're breathing in and that we're all not sick is really, really amazing, right? Okay. So you guys, any questions, just let me know. Make sure you wash your hands before you leave, spray your bench, all right? And I hope you guys can get rested up tonight. Yeah. Questions, you guys?
Oh. I am going to steal it.
You can steal everything you want as long as you bring it back.